Hello everyone, this is Priyanshi. Today we will continue on the lecture that we were studying yesterday, and we will start with the revision and we'll go into deeper topics. We'll also do some previous year questions from NEET. So yeah, let's get started. Right, so we started the chapter called cell cycle and cell division, and as I already have informed, this chapter is very, very important when it comes to NEET, right? Okay. So in order to do a very quick revision, let's look at this picture. What is happening? Something that has happened to all of us. Okay. Something that has happened to, okay, just one sec. This going on. All right. So I was saying we have upgraded this. Okay. Okay, got it. Got it. All right. Yes. So I was saying we all have gotten wounded at some point of time, right? Let's draw a hypothetical person who has been injured. Okay, so they have updated these. So okay, what is that means for that? Okay, let's say this is somebody's hand. Okay, and they just got wounded. This is disappearing. Is this also disappearing? This is not disappearing. Okay, so let's say this is somebody's hand, and they just got wounded. So now suppose they have a cut here. Either way, they have a cut here. Yeah. So what would happen is, after some time, this cut would heal, right? Automatically on its own without putting any effort, it would heal. So what happens, let's say this is the cut area. Okay. So how does the wound heal? It heals because the cells here start dividing, right? The cells start dividing and they start making new cells to fill up this gap, right? And this happens only due to cell cycle. What is cell cycle? Cell cycle is a series of events that a cell has to undergo in order to make babies, right? In order to divide, right? Don't write make babies, in order to divide. Okay, moving on. Moving on. Okay, this is it. I will have to erase this. They have changed this UI. Okay, right. What was I saying? We were talking about cell cycle, and yesterday we studied that the cell cycle, it depends. The duration of cell cycle depends based on what species it is, and even in the same species, different cells will have different speed of cell cycles, right? Because of that, because of that, we divided cell cycle into two phases, right? The first phase is called interphase. Interphase. What is interphase? Interphase is the gap between subsequent divisions, right? Between two M phases. Second is M phase. Interphase is also called resting phase. It's not that cell is not doing anything there. Cell actually does quite a lot, but it is not as dramatic as the cell would be in M phase. We'll see what happens in M phase in subsequent lectures. First, let's just look at interphase and see what happens in interphase. We saw that in the interphase, because it's very, very long, right? Cell spends 95% of its time in interphase, right? Because of that, interphase is also divided into stages. What stages? Interphase is divided into 3 stages, right? G1, gap 1, S phase, and then G2 phase. G1, the first gap is when you are increasing your cytoplasmic content. You are also dividing your organelles because you have to make babies. The cell has to make babies. So both the daughters should get cytoplasm, should get their own organelles, right? So that happens in G1. In S stands for synthesis. G is for gap 1. S is for synthesis phase. So what are you synthesizing here? The pores of the cell. What is the single most, uh, the how I put it, the most important cell organelle which controls the entire cell? DNA, right? DNA, nucleus, है ना? है ना? DNA. So what are you doing? You are actually replicating this DNA because you want to make sure that this DNA is divided equally between two daughter cells, right? So this DNA could also be divided. For that, you need to have another copy of the DNA because you want to make sure that both the daughters get the same number of chromosomes that the mummy cell has, right? Okay. What happens in G2? In G2, this is what we will study today. What happens in G2 is what we will study today. Okay. M phase is also divided into two stages. Okay, first is when the nucleus will divide. This is called karyokinesis. Still, we have to cover this also in later phases. Subsequent lectures, karyokinesis and what else? Cytokinesis. Karyokinesis, as we will study, is also divided into four phases. And let's just do the names. We'll get more familiar with them over time. Prophase, metaphase, anaphase, and telophase. Don't worry about these. If the names seem weird, it's okay. You will get used to listening to these because we'll talk about them so many times. Okay. Yes. So cell cycle, we were studying cell cycle. We were talking about a cell that has decided to make babies. In order to make babies, it first had to undergo interphase and G1 of interphase, right? Right. Okay. Let's do a very quick revision of what the cell did when it entered G1, right? How do I erase everything? Clear all. Yes. G1 phase. G1 is the first phase of interphase. Now, whenever a cell has decided to make babies, it has to enter G1 phase, right? Cell here grows a lot, which means that the cell is metabolically active. Also, this is the phase which is most variable in duration. Cell can take anywhere between 6 to 12 hours when it's in G1 phase. Why? Why is the duration so variable? There's a very good reason for that. Thing is, here the cell becomes smaller in size. When cell is born, it's a baby cell, isn't it? We have a baby cell here. We have a tiny little cute baby cell. Now this cell wants to divide, So in order to divide, it first has to grow in size and become a strong, mature cell, right? Mature cell. In order to become a mature cell, it needs a lot of nutrients. It needs a lot of nutrition. How will it gather nutrition? It will gather it from the medium outside, right? It will search for food, obtain it, and that's when it will have nutrition. After getting nutrition, this cell would be able to divide its cytoplasm, right, among the daughters. So this has to happen. This happens in G1 phase. Okay. Yes, there grows in size. Duration is variable because it depends on the amount of. It depends on the availability of nutrient in the surrounding. What do you call it? Surrounding environment, right? Okay. Moving on. Cytoplasm increases, like you already have drawn here. Okay. Most organelles, like ER, Golgi, all of these also double here. All right. Chromosome number. What is the chromosome number in G1? See, in any cell which is in G1, it's simply growing, right? It is simply increasing in size. So nothing happens to the chromosomes. Chromosome number stays the same as you would see in a normal somatic cell, which is 2n. Right? In all of the cells, chromosome number will be 2n. Now 2n for humans is 46. Okay. Okay. Why 46? Why 2n? It's because half of your chromosomes are from your mom, half are from dad. Okay. N number of chromosomes from mom, n number of chromosomes from dad. So when we write them combined, we write 2n. Okay. 2n for you, for me, for all the humans is 46. Okay. 20 23 chromosomes from mom, 23 from dad, which adds up to 46. So all of us have 46 chromosomes. So where is it? Chromosome number here will be 2n. What about the DNA content? DNA content is also the same as you would find in a normal somatic cell, no matter if it is dividing or not. Okay, we write the normal DNA content as 2C. Okay, chromosome number is 2n, DNA content is 2C. Okay, then the cell is like, okay, now I have grown enough and I'm very big, very strong. Now I am ready to proceed further in the cell cycle, and to do that, it has to enter which phase? After G1 comes stitching. Synthesizing what? Synthesizing DNA. S phase. Synthesis phase. Synthesis of what? Synthesis of DNA. Right. Cool. Then let's go to the next phase. S phase. In S phase, what happens? DNA doubles. DNA content doubles. So, okay, let's say if so we have 46 chromosomes, right? Let me draw these 46 chromosomes. I'm only drawing 4, but we have 46 chromosomes, right? So this is how they normally look. You know? But this cell is very determined to make babies, right? In order to make babies, it has to divide. Like it has to make sure that both the daughters get 46 chromosomes. Right? We cannot do like one daughter gets only 23, the other daughter gets 11 and a half. We can't do that. We have to divide it equally. To divide it equally, the number one thing that it has to do is to copy its DNA. You know? So what it does is, it would create, let's say this is the DNA here. You know? This is the DNA here. It would create another photocopy of the same DNA here. Okay? And of course, there'll be proteins and stuff. Now, what is this? What is the structure now? This is also a chromosome, right? Is this a single chromosome or is this a single chromosome? The answer is both are a single chromosome. Okay, how do we know? Because beta, they have only 1 centromere. Both of them have only 1 centromere. How do we count the number of chromosomes? Very simple. If somebody tells you we count the number of chromosomes, you only have to look at how many centromeres can you spot in that picture. We don't care how many of these arms are there. We don't care at all. We only care how many centromeres. You know? So these are four chromosomes. You know, this is 1 chromosome. This is also 1 chromosome. Okay. What is the difference there? They obviously look different, right? This arm of a chromosome is called what? Chromatid. Chromatid. Okay. And one here, this is 1 chromosome with only 1 chromatid. What about this guy? This is 1 chromosome with 2 chromatids. 1 chromosome with 2 chromatids. And both the chromatids are exactly the same. Why exactly the same? Because we created a photocopy of this DNA, right? We created a photocopy of this DNA. So both the chromatids, no, they carry the exact same gene, exactly the same gene. It is direct photocopy. Okay, exactly the same. So because both of these chromatids are exactly the same, we call them what? We call them sister chromatids. Sister chromatid. And if you don't get it, don't worry. We will keep coming back to this concept. Okay. The very reason why I'm repeating this again and again is because this concept is very new. Right. And very important when it comes to understanding cell cycle, when it comes to understanding mitosis and meiosis. So this is one concept that I cannot afford to let slip from your brain. Okay. So we have to keep repeating this, which I will do. So you just have to listen. So what was I saying? In S phase, the DNA doubles. From 1 chromatid, you have 2 chromatids in the same chromosome. So of course, this has doubled, right? This has doubled. From 2C, you have 4C. But what about the chromosome number? Chromatids have doubled, but chromosome number remains the same, right? Although it has doubled, wait. Okay. Although this this chromosome has double the amount of DNA of this chromosome, even then, it is still a single chromosome, right? It is a single chromosome. So DNA content, what do we write about DNA content? We write that, okay, we write that chromosome number stays the same. Okay. Just a sec. I am facing a bit of technical difficulty. Okay. Did I erase everything? Let it be. Let it be. It's okay. So here in SS, what has happened? Chromosome number has stayed the same. Chromosome number is still what? 12. But what has changed? The DNA content has changed. Right. So we say DNA content is now 4C. And it has changed from 2C to 4C. Now this happens like this transition, this change happens in X-rays. Okay. Also another thing happens is that centriole or centrosome divides in the cytoplasm. What is this? What is this? Hello, let's see what this is. Now, S-phase in nucleus from one thread of DNA, you form two threads, right? But they'll be part of the same chromosome. Number of chromosomes does not increase. Okay. Next thing that happens is in the cytoplasm, you see this happening. You see what happening? You see this happening. What is this? One, okay, come again, bro. Start. This divides into two, right? One centrosome forms two centrosomes, right? So from two, I'm not going to complicate this. From one centrosome, we have two centrosomes. That's all. That is all. Okay. But why did we do this? What is the need for this? For that, let's look at this. See? What do you see right here? These are the centrosomes. Okay. What are these centrosomes doing? They are forming these threads, right? These threads. And these threads will then pull away the two chromosomes. Yeah, actually, this is from meiosis. So here they are pulling away chromosomes. But in mitosis, they pull away the two sister chromatids. We'll see that, don't worry. But the important thing is that centrosomes, okay, also centrosomes occur only in animal cells. In animal cells, centrosome has a very important role when it comes to forming these lines. What are these lines? These are called spindle fibers. Okay, what are these lines called? They are called spindle fibers. What do spindle fibers do? Spindle fibers going to pull away. See, we made this, right? This was one chromosome. We photocopied the DNA, and now this one chromosome looks like this, right? Right? DNA, one sec, DNA here and DNA here, exactly the same, right? And so now this is prepared, right? We have two photocopies of the same DNA, but they are still together. They're still together. They're still in one chromosome. They're still in one chromosome. We don't want this. We want it to be two chromosomes. So we can give one chromosome to one daughter and second chromosome to the second daughter. Right. So what do we do for that? We pull these away. We have to pull these away. Who will pull these away? Spindle fibers. Spindle fibers are going to pull these away from each other. All right. Okay. So they pull these away using spindle fibers. What are these spindle fibers? Spindle fibers. What are spindle fibers made up of? They are made up of tubulin. What is tubulin? Tubulin is also a protein. But where did we get this tubulin from? This happens later. This happens in M phase. Okay, but We have to do preparation for M phase, right? Before beginning M phase. So that preparation, what preparation? Making this tubular fiber has to start right now. Right? In S phase? No, not in S phase, but in the next phase. What is the next phase? Next phase is the G2 phase. Second chem. Okay. What happens in G2 phase? In G2 phase, cell continues to divide. It continues to grow in size, but along with it, it synthesizes a lot of protein. See, a lot of protein. Tubulin protein is also needed here. Okay, tubulin protein was synthesized here. What else? For packaging DNA, we synthesize chromatin, histone protein earlier. Now we need non-histone chromosomal protein. So we need a lot of proteins to be synthesized here. Okay. Also RNA. Why RNA? Because you know the story, right? From DNA, we get RNA, and in this RNA forms protein. Right? So if protein to be formed, we must have RNA. Right? So RNA proteins both will be synthesized. Okay. Once this is done, once we have a lot of proteins, once we have a lot of enzymes for M phase, now the cell is prepared. Cell is like, okay, I am ready. Now I will become a mommy. Okay, I am now prepared to become a mommy. Right? See here. So many enzymes. Now so many enzymes have been synthesized, and now the cell is like, okay, I am ready to rock. I am ready to make babies. Okay. So now this will happen. What about the chromosome number here? Chromosome number here stays. Wait. Oh my god, I got confused. Chromosome number here stays the same, which is 2n. DNA content is also 4a. We did not do anything with chromosome or DNA, right? So it is the same as it was supposed to be at the end of S phase, right? We must remember this. If somebody asks what is the number of chromosome in human cell, which is in G2 phase, they'll say it is the same as it was in G1 phase. It is still 46. But if someone asks what is the number of chromatids, chromosome will look like this, right? So one chromosome has, like we have in total 46 chromosomes, but one chromosome has 2 chromatids, right? Then what would 46 chromosomes have? 46 chromosomes would have 46 into 2, which would be 70, 90, 92. Yes, 92 chromatids. So many. Right, chalo, okay, anyway, let's move ahead. So this table, important. I saw this from another teacher who teaches on YouTube, and I found it really informative. So let's do this. What are we doing here? We are only taking a note of what is the DNA content in different cell cycle or in different phases of cell cycle. Okay. Okay. So we start with G1. In G1, we start with the DNA content is normal in G1, right? It is here. It is at 2C. 2C DNA content. Okay. But when we go to S phase, what do? At S phase, we have to double the amount of DNA, right? We have to double the amount of DNA. So we increase it from 2C to 4C, right? So we go from here to here in S phase. Okay. In S phase, DNA goes from 2C to 4C. Okay. In G2, we don't do anything with the amount of DNA. So it stays at 4C. Okay. In G2, it stays at 4C. At G1, it was 2C. At G2, it is 4C. Now what would happen? We have a fully grown cell. Here, right? And what would this cell have to do? This cell has to make babies, right? Smaller babies. So when it makes babies, what would it do? It would divide its DNA among 2 daughters, right? So when this happens, from this cell, from one of these cells to one of these cells, what happens is that is that the DNA content actually gets halved, right? Right? So we will try to that at Interphase. DNA is now again back to what? 2C. DNA change the amount of DNA in one cell changes from 4C to 2C. Earlier it was 4C here, right? But here it will have 4C. 2C, I'm sorry. Here it will be 2C. We want it to be like that because we want all the cells of our body to have the same amount of DNA, don't we? We do, right? Okay. So this is this. I hope there's no doubt in understanding this. If there's any doubt, please comment. Okay, whatever the nature of doubt is, please comment. I will try my best to resolve them quickly. Okay. Alright, moving on. So now so much has happened. We've seen so much happening in interface. What has happened? What has happened? We saw a baby cell growing into a strong mature cell, which is ready to now make its own babies. Right? And it had to go a lot. One thing I forgot to tell you is that in G2 phase, we saw that most of the cell organelles would replicate in G1. And ER, Golgi, all this gets replicated in G1. But in G2 also there are some organelles, cell organelles which replicate. What cell organelles? The cell organelles which are semi-autonomous. What are the semi-autonomous cell organelles? The cell organelles which have their own DNA. Which organelles? Basically mitochondria and blastocysts. They will replicate in G2. Okay. These cell organelles replicate in G2. Rest everything replicates in G1. Okay. Alright. Okay, but after all this is done and dusted, now the E cell would know when to bud and divide. See, look, it starts with with a very small cell, right? Now it has grown in size. After it has grown in size, after it has doubled its DNA, now it is prepared to split into two. Right? It actually doesn't split, it does budding, right? Which means that it grows a bud on top and it passes half the DNA to that bud, right? Right? Yes. Okay. Cool then. Let's move ahead. Okay. Good question. Very good question. We are considering a hypothetical plant, and in this plant, 2n equal to 40. Like for humans, 2n is what? 46. So for this plant, 2n is equal to 40. Now they are asking what would be the number of chromosomes in a pollen grain. So we know that in all the gametes, for humans it is what? It is sperm cell and egg cell. Now for plants, what is the gamete? Gamete is pollen grain. Oh, okay. So supposedly the smart pen. Not. I guess I'll have to get used to it later. I don't know if I use this now. Let me use the normal pen. Okay. Yes. So what was I saying? I was saying that the NEET has only half the number of chromosomes compared to what the normal somatic cells do. So in human cells, let's say for humans, normal cell, let's say if I take a cell from your skin, from your inner cheek, from your gut, wherever I take a cell from, in any cell I will find 46 chromosomes. Okay, but if I take your gamete, okay, if I take a cell from if I take a germ cell, okay, let's call it. If I take one sperm cell or if I take an egg cell and I try to study what is the number of chromosomes inside these, then I will find that it is not 46, it is actually half of that, it is actually 28. Okay. Same happens for plants. For plants also, plants don't make any sperm cells, they make pollen grains, right? So in pollen grains, what would be the number of chromosomes? Here it is. I should have actually written male in it. Why am I saying this? This is something that we will study when we study sexual reproduction in flowering plants. Okay. So yes, number of chromosomes in the male gamete would be half of 40. You know, if 2n equal to 40, then n would be equal to 20. Easy. Number of chromosomes in G1 and G2. We know that in G1 and G2, number of chromosomes does not change. Number of chromosomes does not change, so it will stay 40. Right? Okay. Number of chromatids. Okay. Now they are asking the real thing. Number of chromatids. So in G1, chromosomes look like this. You know, there are 40 chromosomes and all of them look like this. In G2, what happens? So we say 40 chromosomes and 40 chromatids. Cool. But in G2, what would happen? This one side. In G2, what would happen? This would transform to this, right? This. We'll have 40 chromosomes like this. And so now we see 40 chromosomes, yes, because the number of centromeres, rest of centromeres, number of centromeres is still the same, you know, right? But what has changed? The amount of the number of chromatids, you know, we have sister chromatids now, right? Both of them are sister chromatids. Here it was the only child. Now this child has a sibling, right? It has a sister. Both of these are sister chromatids, you know? And in one chromosome we have two chromatids. So here we have 40 chromosomes. But we have 80 chromatid, right? 80 chromatids, right? So number of chromatids in G1 is actually the same, 40 as a number of chromosomes. But in G2, the number of chromatids is 80. Right? Okay. If the DNA content in a somatic cell is a normal cell which is not gametic, is 2C. What would it be in G1 and G2? In G1, there is no doubling of DNA that has happened. So in G1, it will still be 2C. 2C. But in SS, we know that the DNA content changes from what? Changes from 2C to 4C. So a cell which has entered G2, it means that it has 4C DNA content. So we write this. So we have to write that in G2 it would be 4C. In G1 it would be 2C. Number of chromosomes in polymerase would be 20. Number of chromosomes in G1 and G2 would be 40. Right? Both 40, 40. Number of chromatid changes 40 and 80. Which part of this is not clear? If you understand this, half the story is done for you. You don't have to worry about this chapter anymore. Everything is easy from here. So I hope this is also clear. Okay, now we'll move forward. Yeah, this is about the time duration that each phase takes. You know? For humans, a human cell which is grown in culture cell. If a human cell is grown in culture outside, then it takes 24 hours to divide. You know, if a cell is, let's say, a petri plate, which is nothing but a plate, okay, plate made up of glass. We pour in some food for the cells to be grown on this plate, and now we have kept cells. So these are the cells, and we are stimulating them. We are giving them factors that should promote their division. So we are like, hey, divide. We are like continuously pinching, divide, make babies. So if we do that, then if I start at, let's say, 12 p.m., I start telling them, make babies, then it will make babies. But when? At 12 p.m. the next day. Right? Give me a sec, my laptop's battery is low. This one. Just a sec. Yes. So this cell has made a baby after 24 hours. Out of these 24 hours, no, 23 hours were spent only in preparing for dividing. Okay, and the actual division happens in the last hour. Okay, in the last hour. All right. So till 11 p.m. the cell was still preparing. Yeah, till 11 p.m. it was still preparing. Only one hour it actually divided. So and this is only 1 hour. Okay. G1, like I told you, most variable, it takes between 6 to 12 hours. DNA is doing, right? In S phase, cell takes 6 to 8 hours. Anywhere in between depends on what type of cell it is. Okay. Here the DNA content would double from 2C to 4C, right? In G2, less number of hours, right? Here the DNA content is 4C already. They have represented with N, but I don't think this is correct because people might confuse it with the Ploidy. Right? Okay. Yes, and in M phase what happens? This 4C gets divided into two cells. So both the cells have 2C, 2C. Right? Right. And only one is taken in doing that. Oh yeah. Now let's read about a very weird phase. Very weird phase called G0 phase. See. Hmm. Let's say this was a cell. In a normal cell cycle, what do we do? We say this is a baby cell. Okay. This baby will grow, right? Grow, and become a bigger cell. And then it can finally divide to form two cells. Right? Then what would happen? This daughter would again grow, reach here, and then divide again and make babies. But what if a cell doesn't want to do this? It is like, nah, I am done. I don't want to make babies anymore. Then what options does it have? It has two options. It has two options. Number one, it has two options. Option number one, option number one is to differentiate. Differentiate and become a permanent cell. Permanent cell. So it can become a neuron. It can become a skin cell, epidermal cell. It can become what? It can become a muscular cell. It can become a permanent cell. This is one option. But what if the cell is like, no, I don't want to become a permanent cell. I want to divide, but not right now. I want to act as like, let's say, reserve material. So if some normal cell, if a permanent cell is damaged, then to replace that cell, I am available. If I am needed, then only I will pick up my cloak and I will play the role of the superhero, right? And save this human, right? But that result, when a cell does this, we say that the cell has entered a G0 phase, or we call it quiescence stage or resting phase. This interphase, the entire interphase is also called resting phase. So yes, it can enter a G0 phase. When would this happen? This would happen only, babies cannot do this. When this baby has grown in size, at least somewhat, then only it can enter, it can decide, okay, I have to enter G0 phase. Okay, this will become clearer as we move further. This cell can exit the cell cycle in two ways. It can become a differentiated cell, like you see a neuron, or it can become, or it can be in a quiescent stage, which is reversible. Cell which is in quiescence state or G0 state can enter the cell cycle again, can start making babies if there is a need to do so. Cell in G0 phase can re-enter the cell cycle. Cell can exit cell cycle and become either a differentiated cell or become a quiescent cell and enter G0. Is this clear? I don't think there's anything to note. Be clear. With this stage is reversible and the cell can re-enter the cell cycle. Okay. Why do we do this? Because yeah, cell division is energetically very expensive. It is not easy. Right? Making babies is not easy. Everyone knows that. All the humans know that making babies is not easy. Is it? It is not. You have to, for women, they have to carry the baby in the womb for so many months, right? Even after that, they have to nourish the baby. So it is very energy extensive, intensive process, right? So the cell is also like this, right? Cell is also like, I can't become a mommy right now. Like people do, right? They are not ready to become a parent now. Cells can also be like that, right? Cell, even though there's not enough nutrients allowed, even if I make babies, what would my baby eat? So when nutrients are not enough, when the growth factors are not enough, then the cell is like, let me enter G0 instead of dividing and making new babies, because I don't want my babies to live in such a situation. So I will just wait, see if the situation is favorable, see if I'm even needed to make babies. And if I'm needed to make babies, then I will make babies. Right? Okay. Okay. And what is the advantage of this? The advantage is that this is a stem cell. Yeah? Stem cell is a cell which is mommy of all the cells in our body. Right? If all the stem cells become permanent cells, differentiate and become permanent cells, then we will have no stem cells, right? We will have no stem cells at all in our body. That is not what we want. We want stem cells to stay alive, right? So what would a stem cell do? Stem cell will divide. You know? Some of these can become neurons, can differentiate and become neurons. Some of them will stay here, but not divide. Okay, they're there, but they're not dividing. These are the cells in Interphase. So they can divide, they can differentiate when they are needed. Okay. I think it is clear, right? So let's move on then. Interphase is a non-dividing differentiated stage. In G0, what is this? Arrested in G. Okay, let's some cells after they enter G0, they can never wait. I don't think we should. Okay, what do you see right here? Tell me. Let's take a quick break. Tell me how many chromosomes you see and how many chromatids you see. Count fast. Pause the video. Start counting. How many chromosomes? 46. Okay, wait, there is something wrong here. These are not joined, see? These are not joined. These are not joined, right? So how many chromatids? How many chromatids? 46 chromatids. These are 46 chromosomes and these are 46 chromatids. Okay, alright. This one, alright. It's a very weird term that I came across when I was preparing for this lecture. We'll do this later actually. Let's solve a few questions and wrap up this session. Okay, we'll do this later. We'll do a mitosis later. Identify the correct statement regarding G1 of interphase. Okay. What happens in G1? DNA synthesis or replication takes place. No. It happens in which phase? S phase, right? Reorganization of all cell components. It is not that dramatic, right? Cell is metabolically active, grows, but does not duplicate in DNA. This is right. Nuclear division obviously doesn't take place. So the correct option is option number C. Some dividing cells exit the cell cycle and enter a vegetative inactive state. This is called G0 stage. Okay. This process occurs at the end of did I tell this? They can enter G0 only from G1 phase. Okay. Only from G1. Now, why? Because once a cell has doubled its DNA, once the cell has started doubling its DNA, then it has to divide. It must divide. Right? See, if you have wheat flour at home, If it is just there in a container, then you can do whatever you want with the wheat flour. You can make cookies with it. You can make a cake with it. You can make chapati with it. But if you have made a dough out of this wood flour, okay, wood flour, wheat flour, if I do you, if you had already made a dough out of it, now you have to make chapatis, right? If you have already made the dough, now you have to make chapatis. You cannot let it lie like that. It's exactly like this. If you have started doubling your DNA, now you have to make babies. You have to make two sets. You cannot stay with double the amount of DNA. No, not allowed. Not allowed. Against the rules of your right. So yes, if cell has started dividing the DNA, then it has to enter even. Okay. It has to enter cell cycle and divide. Make babies. Okay. So if you have to exit the cell cycle, the only way you can exit is from G1. The only way you can exit a cell cycle is when you are in G1. If you enter S phase, then you gone. You have to divide. No escape. Okay. I'll later. So back to the question. Process occurs at the end of G1 phase. Okay, after the cell is grown up, cell is like, okay, now I want to stop dividing. Only a grown cell can take a decision on that. Okay. Cell in G0 phase, exit the cell cycle. We just saw that. Right? Correct sequence of phases in cell cycle is gap 1, then S, then gap 2, and then M. Whichever option is G0. Right? This is the right option, right? Okay. See, such stupid question. Not stupid, but easy, right? This is actually NEET 2019, so it's very recent also. During cell growth, DNA synthesis takes place in synthesis, S phase. Easy. Somatic cell that has just completed the S phase of its cell cycle as compared to the gamete of the same species has. Okay. So, like, let's say this is a sperm cell. Now, sperm cell will have 23 chromosomes, right? And a normal somatic cell has, for humans, 46. This is not very good. Right? Sperm cell has 23 chromosomes. Normal cell, let's say a skin cell, has how many chromosomes? 46 chromosomes. Okay. Now this, but okay, we're not talking about a skin cell, we're talking about a cell which is dividing. So it has entered S phase. Even in S phase, the chromosome number stays the same. Right? 4 times what? So it has twice the number of chromosomes. Somatic cell has twice the number of chromosomes. Yes. Yes. 4 times the amount of DNA. 4 times the amount of DNA. Okay. This actually the options are wrong. It is no. Twice the amount of chromosomes is it should be actually 8 times the amount of DNA. Right? Because it is an S phase. It has completed S phase. Which means that the DNA content is actually 4C here. Right? In a gamete, the DNA content is only C, 1C. Okay, no, actually it's right. It is right. It is right. It should be this. Yes. I'm sorry, my bad, I got confused. So this option is correct. This option is, oh, I can do this, okay. No, I can't do this. Option is correct. During which phase of the cell cycle, amount of DNA in the cell remains at 4C level if the initial amount is 2C. You know this. You know this. We drew that graph, right? 2C to 4C, then 4C, then right? So it stays constant in which phase? In which phase? In only G1 and G2. No? It stays at constant at 2C in G1 and it stays constant at 4C in G2. So it is only G2. Alright, moving on. In S phase of the cell cycle, amount of DNA doubles in each cell. Yes. No. Let's look at all options. This happens is amount of DNA remains the same. No. It doubles. Chromosome numbers increase. No. The amount of DNA increases, but the chromosome number stays the same. Amount of DNA is reduced to half more weight. So option is A. Given below the schematic breakup of the phases of cell cycle, which one of the following is correct indication of the stage and phases of the cell cycle? Okay, let's see. What is A? This entire thing is interphase, right? This is Z1, this is S. So D is actually S phase, right? This is true, right? Okay. For this, no, we have to read mitosis also. This is karyokinesis, C is karyokinesis, A is cytokinesis, okay, and this is C and A. This is B is actually anaphase. But okay, no problem. Here the answer is actually what? D synthesis phase, right? Right? Okay. Now let's move on. At what stage of the cell cycle are histone proteins synthesized in eukaryotic cell? Histone is synthesized in. So protein synthesis happens in G1 also and G2 also. But you need histone to synthesize DNA, to duplicate DNA, right? And for packaging DNA, you need other proteins also, but for duplicating, for synthesizing DNA, you need histone. Okay? So we are going to duplicate DNA in S phase. So we prepare for S phase in G1 phase. Okay? So in G1 is when you wait. Let's check this question. Surface definitely not. Then it should be only S phase. No, but it is synthesized in G1 also. I will check this, double check this, but it happens in G1 and G2. Alright. Next question. In a somatic cell cycle, in G1 phase, DNA content is double the amount of in a somatic cell cycle. G1 phase, DNA content is double the amount of DNA present at the original cell. DNA application takes place in S phase. See, so many questions are asked on this. S phase, right? A short interphase. No, interphase is actually very long, and mitotic phase is very short. G2 phase follows mitotic phase. Oh, mitotic follows G2. First is G2 and then it is interphase, right? Okay. So we are done with questions asked on interphase. Now we have to read the second part, which is mitosis, which is something that we will do in the next class. So I hope today you enjoyed today's session. If there are any doubts, please comment below. I will address them promptly. And I'll see you in the next class, which is tomorrow at 8 p.m. So I'll see you there. Bye.